I will also have the key available up front, so if you miss anything, you can come up and look at it. The first question, apoenzymes are inactive by themselves, and they must be activated by a cofactor. Good. Now, I could word that question differently. Apoenzymes and cofactors together make a what? Holoenzyme. Holoenzyme. Good. You got it. The whole. Which of the following is not created in aerobic respiration? That was a tricky one, right? We need oxygen, but we don't create it. We need it, but we don't create it. Next, lipids are generally processed in the Krebs cycle. So lipids, proteins, we talked about that on the board. We had a picture of it too. They're in the Krebs cycle. Number 4, which of the following use chemical bonds for their energy source and organic bonds for their carbon? That would be B, chemoheterotrophs. Number 5, which of the following statements regarding the ED pathway is true? It would be D. This pathway creates one net ATP. How many NADH does it make? 1. Good. How many pyruvic acids? 2. Excellent. The term aerotolerant anaerobe refers to an organism that A, does not use oxygen but is not harmed by it. It tolerates it. It doesn't matter one way or another if the oxygen is present. Number 7, almost all pathogens are chemoheterotrophs, just like us. Number 8, energy is released in glycolysis due to glucose oxidation. So remember, oxidation means we're losing electrons. The glucose is giving its electrons removed. Number 9, the biosafety level for a clinical microbiology laboratory working with airborne pathogens such as TB, that was a big hint with TB, that's a BSL-3, so BSL-3. Going back to number 8 really quickly, probably the next, so most people got it, but probably the one that most people put in the wrong place was glucose phosphorylation. So be careful with that. If I'm phosphorylating glucose, what does that mean is happening? I'm adding phosphate to glucose. Does that give me energy? No, it requires energy, right? Now, if I have dephosphorylation, that would give me energy. So you have to be careful. So phosphorylation means I'm putting it on. Dephosphorylation means I'm taking it off. So if I'm putting it on, I'm losing ATP. That's the opposite of what I want. Just wanted to clarify that. Number 10, the following pairs of microbes that were mismatched. It was B, the cyclophiles. Number 11, culture medium. So the only information I gave you was it has agar, sugar, and beef extract. Based on those 3 ingredients, the only thing we can assume is it's complex. We know it's complex because it has extract. There's nothing in there that says that it has dyes or salts or anything to make it selective or differential. So the only thing I could go with was complex. Number 12, a microbe that must live in high salt or high osmotic pressure. I was looking for obligate halophile. A lot of people just put halophile, so I took it. There's a safety in numbers, plus I'm not going to ding you for that. I was happy you had halophile. What is the first product made in Krebs? That was citric acid. Number 14, enzymes increase the activation energy. That's false. Remember, they decrease activation energy, they increase reaction. Number 15, it is true that bacteria can convert gaseous nitrogen. Okay, then we got to 16 and 17. These are fill in the blanks. So I'll just read the answers on each line. For A, it should be cytoplasm. For B, mitochondria. For C, mitochondria. And some people said inner mitochondria, mitochondrial matrix. That was great. If you just put mitochondria, I'm happy. D is cytoplasm. E is cytoplasm. And F is the plasma membrane. Number 17, going across again. A is 2. B is 2. C is 2. D is 6. E is 2. F is 3. G is 2, H is 34, and I is 36. And the last extra credit question, that was the binary fission. That's how they're going to reproduce this through binary fission. So hopefully those make sense. Will you see them again on the exam? Yeah, not the exact same questions, different varieties. But make sure if there's a term you struggled with or a concept you struggled with, take some time to review it and be sure to ask me. Okay, I'm here to help you, so be sure to ask. Your study guide is posted, so you can go in and look at it. We do have something more pressing though coming up next week. What do we have next week? Lab exam, yeah. So I've had some students asking me like, how do I study for the lab exam? This right here is what I recommend. It is over labs 8 through 12, which doesn't sound like a lot. It's like, oh, 8 through 12, I can do that. I will say 8, pretty easy. That one's plates, broths, slants, knowing when to use each one. That's pretty easy. That's pretty straightforward. It does go into the different types of media, but it's pretty easy. Okay, but then we get into the meat, 9, 10, 11, and 12. So I recommend studying these as bite-sized pieces. Think exoenzyme. So what you're going to do, what I would do, this is how I did it as a student. I would go in and I would make either a flashcard, or actually what I liked was to get a big poster board. You know, cheap poster board, piece of cardboard. Heck, you can use an Amazon box, open it up and write inside of it. Write in phenylalanine deaminase slant. Write in the organisms you set up on it. You're going to write what the positive looked like, what the negative looked like. You're going to write what that was looking for, what was the purpose of the test. And you're going to do that process for every single one. All right? If you make it bite-sized, it is totally doable. Trust me, I've seen students in the past. The ones that come in who have studied and broken it apart, they don't feel overwhelmed and they do really good. If you're waiting till the night before and you're trying to read this, your organisms are going to get way too jumbled. All right? So keep it bite-sized. Things to know, the names of the media. When I'm asking you questions on the quiz, I'll say, in my OF media, what does a yellow reaction mean? Or I can say in my OF media, who showed a positive reaction? What color was this positive reaction? So I can ask these questions a bunch of different ways. So make sure you know the types of the media, who was set up, positives and negatives, and what that meant. Knowing the names of the biomolecules that we're looking for, the enzymes, and the reagents involved. Reagents meaning, did we have to add anything extra? For example, with the starch plate, what did I have to add?
Grams iodine.
Grams iodine, yeah, exactly. So just knowing how to read these things out is going to be key. The enzymes that we were looking for. Is the media selective or differential? That's only relating to lab 9. I'm not going to ask you about selective or differential in these. I'm not worried about it. And then again, just making sure you do bite-sized pieces. Okay, if you start now, it is doable. I promise it is doable. All right, and believe me, I've been in your shoes. I get it. I totally get it. I had an instructor who they would just have the tubes out with no names. And they would say, go read this. What reagent is it? What are the positives? What are the negatives? I don't do that to you. And you had 1 minute. You had 60 seconds at each station. Stress. Talk about endospores. I was hatching it as we walked. You know, I was like, oh my gosh, yeah. So I'm just saying this setup is not to like punish. It's just to make sure you know the material. Okay, is everybody good with that? We know what we're studying? Okay, and I'm here. I know lab will end early today, but at the same time, I'll keep the lab door open until noon. So if you leave, come back. That's fine. You know, you can do that. I'll leave the door open. I also have lab tomorrow from 12 to 2:20 next door. If you have a quick question, drop by. That's fine too. If you can't make it to my office hours, I'm here for you. Just let me know. Okay, so let's go ahead and start going through our results. Let's finish up the carbohydrate lab. That was lab 12. So we're going to read out our MRVPs from last time. So we had incubated those, and I said, yeah, let's incubate them a little bit longer. So what we're going to want to do with these guys is set up our reactions. Now, looking at our MRVPs right now, I can't tell a difference. These are the little tubes with the screw caps. I can't tell a difference just by looking at them, who was positive, who was negative. So we need to do some additional things. The first thing we need to do is I need to aliquot my bacteria into a blank tube, because we're going to run 2 different tests on the same bacteria. So I'm going to borrow yours for a second. So your E. coli tube, you're going to aliquot a mill. Everybody has a pipette into this blank tube. All right, and that's going to be your E. coli. And then your lab partner is going to do the same thing for the Klebsiella. All right, so when we're done, we're going to have 2 tubes for each organism. Now, before you aliquot, make sure you tight cap and mix them. I'm going to put it in version. So go ahead and do your aliquot now. Everybody should have a pipette at your desk. I'll put scissors out. Sometimes these pipettes, I swear, they're hard to open.
So for 81, we're doing our
I'll put scissors out.
MRVP. And then
Close the back door so I'm not shouting all that. So sorry, sit down. So aliquot first. Don't do anything else, just aliquot. So aliquot means to take a mill of your bacteria and put it into the fresh foods that you have. And I would recommend keeping track of who's doing what. So have E. coli on one side, Kleb on the other. Because we're not labeling them, so just keep them together.
Monday
Sorry, I'm gonna steal it if they don't do it. Don't forget.
we have to figure which one is going to because we have to put MR or we put both. So we don't know which one.
Alright, so once we're done with the aliquot, we'll go on to the next step together.
So,
Hold it. I need to go up to that. This is lab 11 now.
no, this is from lab 10. Or lab 12, sorry. So we're just going to add...
And then once you're done with your aliquot, you can throw your pipette away in biohazard, get it out of the way. And it doesn't have to be a perfect mill, just one mill as much as possible.
We're on this page.
Or mill 0.1.
So we're trying to add something.
Bacteria wants
We need
out.
to add, like, it's an aliquot, basically like a reagent, so we want to add it, so we're going to add it to start, to do the results, we need to figure out how to do this.
Okay. Is everybody done with your aliquot? Okay, so once you're done with your aliquot, I want to skip to do the VP reaction. Not VR, VP. Okay, VP reaction. So what I want you to do is I want you to add 12 drops of VP reagent 1 and then 4 drops of VP reagent 2. Now the reagents should be at your desk, so you're going to find the right reagent. Actually, I think you guys have a little bit They're over here.
Which one are we?
Yeah, so here's VP reagent 1 and 2. For that side of the room, you have 1 at each desk. We're like kind of sharing. So go ahead and come over. You're going to do 12 drops of VP1 and then 4 drops of VP2. Make sure you're adding the right reagents. You have lots of reagents at your desk. So you're adding this into the one that you just aliquot to, the 1 mil tube. So 12 drops of VP1 and then 4 drops of VP2.
These are VP.
Yeah, VPA is VP1. Yep.
We don't get it, that's why I'm asking.
If you have A and B, A is 1, B is 2. This is also in your lab book. If you can't read my writing, it's still in your lab book. So it's on page 1, or sorry, page 81. Now you're going to do this to the ones you aliquot. So you're going to have your E. coli, and then your lab partner's going to have their Klebsiella. It's the one that's over on the
No,
side.
they're both MRVP.
Okay, so what do we do?
Unfortunately, we don't have enough reagents to have it.
I will put 1 milliliter.
So 12 drops of A and then 4 drops of B. So on this side of the room, you should have 1 at your desk. So you're going to have to share. It's going to
I'm
go
just trying
around
to
the
figure out what we
desk.
did before.
Yeah, we did this.
All of these, right?
Just this.
And you did
Just this. We did this already.
That
This
was
is the only one we're doing. We didn't finish it last time. Forget about that. We already figured out the results for those.
and these ones we did.
Yeah, no, forget about that. We're only focusing on it.
We already did that.
Right, we did it, but
Okay,
now
so
we're looking
12
at the results.
drops of reagent A for 1, and then 4 drops of reagent B
Like
for
we're trying
2.
to figure this out. That's the other results and figure out.
Now, once you add
We
your
have to
drops,
add a reagent.
tight cap that puppy and shake it. So once you have, and not gentle shaking, we're shaking for a minute. Okay, so as soon as you add those drops, Tight cap, yeah, shake it. Shake what your mama gave you. Shake it, shake it, shake it. Yeah,
I
let's
like
keep
how
going.
what
Shake,
you
shake,
want
shake.
to don't
We're
understand.
doing this with intention. Alright?
Well, you need two. You need two. You need
My dog, when he gets one of his favorite toys, I'll say, shake it, he'll start shaking it. And then he makes himself dizzy, he falls over, and he's like, I'm good. Oh, it should say on there, so EPA, right?
Could it be that one of them is?
We did it right. Well, I don't know what it has.
She said EPB. We don't have it on the label.
Oh.
Can we ask them?
Did you answer it? No, because I'm off.
Okay, shake vigorously. Yes.
I just have a question.
Yeah.
Right there.
We don't know which one it has.
The VP.
So we haven't done the
Right here,
We
Vogue's
haven't done
process.
the first one.
Okay, so we're still in that one. So did you aliquot?
No.
Okay, so you're supposed to aliquot. We were all doing that together into a blank tube.
So we
Okay.
grabbed this.
Yep,
and
so
then
you
add
use
it.
that to take a mill from this into one of your sterile tubes. Do you have a sterile tube? Yep, to one of your sterile tubes. And then you're gonna do the same thing, that's Klebsiella, you're gonna do the same thing for E. coli. So you have 2 tubes each. Then to the one you aliquot to, you're gonna add your VP reagents. Okay, that's where you're gonna add the 12 drops of A and the 4 drops of B.
And then say something.
We're gonna shake them, yeah, so you're gonna add your drops, you're gonna shake them, and then you're going to see what reaction you get.
Okay, can I have the sterile too?
And this is E. coli. She did.
Okay, let's clear it.
We're aliquoting from each organism, so let's go get filled into a container.
Just
Coli
one.
gets in there.
Okay.
And then E. coli. Then once you're done to that mill, you're adding 12 drops of EP1, and then 4 drops of EP2.
Clutch. So we can forget about this because we don't need it.
And that one's getting your post-it, and then you're gonna do the same thing.
The other thing, and
This is one.
And you can tell the reagents will say.
And that's the one that you just-
That's the reagent 2.
Yep.
One, okay.
Alright, so after you shake it for a minute, you're gonna take the cap off and let it sit. I know we never take cap off. We're gonna take the cap off and let it sit. It has to have an oxygen reaction time.
And then we add 12 drops of the BP.
Yeah, same thing.
Okay, so label this one and pour
Good.
it.
See, lots of shaking. Everybody just has to shake it. Are you already doing it? Good job.
Add 12 drops of BP,
Right, to both?
yeah. And 4 drops of BP, 2. Yeah. 12 and 4. 12 drops of BP.
Which one's 12?
Both.
That one?
And then we add the 4 drops of BP, we add the 2.
And then 12 in this one too.
What? 12? Oh, I did 12 already. Is it the
I get that one for you. Thank you. It goes in here.
What's the best?
And we add 4 of that.
And you shake for 1 minute. So at 9:50.
Oh wait, is that?
You need that?
No, I need that.
Oh, you need that.
Yeah. And then that over there is here.
Oh,
Do you have the B? Oh, it's right here. This is B.
Never take
Tiara asked if you would save the results.
Yeah,
She's on her way and she's having car issues, but she'll be
oh yeah,
here.
she just showed up.
Oh, did you? Okay. Oh, there she is. I just got her email. I'm so good at this, aren't I?
I heard you. I was like, I made it.
Okay, did everybody shake? We shook up, we're good? Okay, so now you're gonna take the cap off and let it sit for about 20 minutes. So when we get done talking about everything else, we'll come back to this side. What we're looking for with the VP reaction is we're looking to see if our organism is able to do a reaction in its metabolism process where it's going to create acetylcholine as its end result. Normally, when we do metabolism, we create, through glycolysis, pyruvate as our end result, right? Now, the way we're gonna see if our organisms create pyruvate is to the other tube, the one you haven't had anything to yet. You're gonna add methyl red. So at your desk, you have methyl red, and I had enough for everybody to have methyl red. You're gonna add methyl red. You're gonna add 5 drops of methyl red, and you're gonna see if your tube turns red at the top. So go ahead and do this to your tube that you did not add the VP to. Okay, so go ahead and add your methyl red to the tube that you did not add your VP to. So 5 drops.
So I think one of them, so methyl red. I don't know, I'm feeling good about it. The reactions because I don't know what to do. So red is blue.
You don't have to. You shouldn't do it.
Oh, did you sign it?
You actually can't. You gotta check it.
We have to do the other one and we have to add methyl red.
So you're gonna add your methyl red. Go ahead and just do a couple of gentle shakes. You don't have to do a whole lot. Just a couple of gentle shakes. And we're looking to see which organism is producing a red color. Okay. So did you see an organism producing a bright red color? Yeah. Who's producing the bright red? E. coli. Okay. So E. coli is going to be positive for this test for the MR, for the methyl red test. So he's MR positive. So what does that mean? It means that E. coli is going through normal glycolysis and creating an acid at the end. When we go through glycolysis, what acid do we create? Pyruvic acid. So that's what we're looking for. We're looking for a pH drop. Now, I told you last time, if I'm MR positive, can I be VP positive?
No.
No, yeah, I have to do one or the other pathway. So now that we know E. coli is MR positive, who do we think is going to be VP positive? The Klebsiella. Yeah, we can kind of like eliminate that idea. Now, with the Klebsiella and the VP reaction, I will warn you, it is very weak. The MR reaction is awesome. We get a quick color change. The E. coli is very red. Klebsiella sometimes is a little pink, but then he fades. That's fine. E. coli stays a bright dominant red. That's what we're looking for. With the VP, it's a little more subtle. So we're going to let this sit for a good 20 minutes. We're going to look for a ring at the very top. And it's going to be faint. It's going to turn kind of a tan, peach to pink color. The longer you let it sit, the better, the more of a reaction time you get. When we read these in a medical lab, we let them sit for over an hour. I don't make you guys stay in here for an hour. You can look at it after about 20 minutes. But just know it's going to be a very subtle change. Okay, very subtle. So we will read VP out when I'm done with the rest of the stuff. So just let that sit. Okay, let's go to our protein lab now. So now we're back to lab 12, or sorry, lab 11. Page 73.
She tells us.
Let's start with our phenylalanine plant. We're going to add 4 to 5 drops of ferric chloride to our plant. So at your desk, you should have ferric chloride in one of these white containers. And you're going to find the ferric chloride and add 4 to 5 drops. Now you're going to let that go down the slant, and you can kind of rock it a little to make sure that you're getting enough reagent on there. And you're going to look for a green coloration. So right now they're plain looking. After we add Kovacs, we're going to look for a green coloration. Sorry, not Kovacs. Ferric chloride. Ferric chloride.
11.
So green coloration. So ferric chloride. 4 to 5 drops ferric chloride. and I was like, oh,
I barely made mine this morning.
I didn't have
So
mine
if it's
yet,
gonna
so
be
I'm
green,
dying.
it's positive for us.
I gotta get through it.
So this one's green.
But I'm gonna have an iced coffee.
This one's negative.
Okay, 4 to 5 drops and very good. Then y'all are gonna listen to me and lecture, and I'm gonna be like, ah! So be prepared to tell me straight down and lecture.
So the green one is what?
Do you get a coffee from here?
Proteus
I
vulgaris.
don't really like the coffee
The
filter.
green
They
one.
taste bad.
Yeah.
And there's one
This one's green and it's positive for
I'll
us.
share my nervous story of coffee here. I went over one day and I just wanted a brew, so I said, hey, do
And
you have
then
any
the
brewed
one that's
coffee? And he looked at me and I
negative
seriously
is
said, we
because
don't
it's
have that
yellow.
kind of coffee here. Which makes me wonder, how are they making the other
And this
coffees?
one's
If you don't
Yeah.
have coffee, that means everything's powdered, you know, and I'm like, ugh, I can do that.
positive because it's
Yeah,
reactive
so I think I'm gonna be really
chloride.
bad and do a rare Starbucks.
Yeah,
I rarely go. Yeah, I love it, but I rarely go in one.
Yeah.
But I have given it.
Yeah, I like the one in Pismo Beach. Is it the coffee beach?
They're so good. Coffee
I
beans.
will drive. Ronnie thinks I'm crazy because there are times when I just drive it with.
It's been crazy. They used to have a caramel drink that was amazing, and then they didn't do it anymore.
Yeah.
And I still believe that. Still makes me sad. Okay, so are we seeing a green coloration with our ferric chloride?
Yes.
Yes. Who is turning green with the ferric chloride?
Proteus.
Good. Our Proteus should be doing a nice green color. Okay, well, what does that mean? If I'm turning green, what does that mean? It means that I am positive for acid production. So remember, in protein land, if I'm positive for acid, that means I've removed an amine group. Remember, we said amines mean pH drops, carboxyls mean pH goes up. So this tells me that my Proteus is going to be positive for deaminase. E. coli was not able to break apart the proteins it was given. So that's why we're not seeing any kind of pH change. Everybody with me on that? Okay. We get to go on to the next one, lysine broth. So the lysine broth is a demo, and I do want you to look at it. They were making it in the back room when we were setting up our lab. So the Tuesday, Thursday lab got to set it up. Our lab didn't get to set it up in person, but I did set up a demo so you can look at it. So lysine decarboxylase broth is looking to see if my organism can create and remove a carboxyl group from an amine group. Now, if I remove carboxyl groups, again, my pH goes up. So this is kind of a funny test. So this is what the broth looks like uninoculated. Okay, it kind of has this golden tone. And if I add a bacteria who is unable to break down the broth and break down the proteins that are inside, this one's Proteus vulgaris, I'm going to say that yellow color. So this is uninoculated, this is a negative result. Okay, this is also an anaerobic reaction, so there's oil on top, in case you're wondering about that. But if I'm an organism who can do decarboxylation, which is Klebsiella, so in your book you'll want to circle Klebsiella as positive. If I can do decarboxylation, what's going to happen is I'm going to get a pH shift towards the upside because I'm breaking off carboxyl groups. And then I turn this really pretty purple color. Isn't that pretty? So pretty. Now, as this organism grows, this will get purple and purple and purple. Eventually, though, it's going to run out of amines in here to break apart, and then it'll start eating the sugars, and if it sits too long, it goes back to yellow. So this is a quick reaction. That's why we can't do these over the weekends, because they eat what I give them, and then they go back to whatever else is in there. So I do want you to record this. I have it up here as a demo, but I do want you to know who was positive, who was negative, and what the colors look like. Okay, let's go to our last one that we're doing in here today, which is the MIOD. So I told you we love these in microbiology because it's a big bang for your buck. So go ahead and take your MIOD. The first thing we're going to look for is motility. So if you hold your beak up to the light, what you're looking for is, does your tube look fuzzy, or do you see a distinct line where you inoculate it?
This one.
Okay, so you see some fuzziness, yeah. And then on the other one, do you see just a line where you inoculated? Who is looking fuzzy?
E. coli.
E. coli, yeah. So our E. coli looks fuzzy because he's able to swim. Remember we talked about how E. coli has flagella everywhere? What were those flagella flagella called? Ooh, she's throwing back terms at us. Paratrichus, excellent, yes, A++. Okay, so paratrichus, right, flagella, it's everywhere. So that means that they're able to swim in this media and spread out. Guess who doesn't have flagella? Klebsiella, yeah. So Kleb can only grow where you stuck him in. All right, so he grows right along that slant line, but the surrounding media should be clear. Is that what y'all are seeing? Okay. The next thing we're going to look at is, was our organisms able to break down the amino acid tryptophan? Okay. Tryptophan. Have you ever heard of tryptophan before? We have a holiday coming up that talks about tryptophan. What is it? Thanksgiving, yeah. We hear about tryptophan in relation to turkeys. When you cook a turkey, one of the things it releases is tryptophan. It breaks down tryptophan. And what happens when we break down tryptophan is it releases a gaseous product that is relaxing and soothing to us. That's one of the reasons why when you cook a turkey, you feel kind of like homely, or you feel a little calmer. I don't know, like when I'm running around for Thanksgiving, I always am tempted to just open the oven and okay, good. It doesn't work. It doesn't work very well. But it's supposed to be very subtle. It's supposed to just help you like calm down and breathe. So it's kind of interesting. We're doing a quick check here. I don't want you smelling it. We're going to see if we broke down tryptophan by looking to see if we get any kind of reaction with Kovacs reagent. So at your desk, you have Kovacs reagent. So what you're going to do for this test is you're going to add some Kovacs. And I want to say it's 5 drops, so let me double check because I don't want to lie to you. Let's see. I have to look at my stuff to make sure that I have on here how much Kovacs to add. Why not say? Add 4 to 5 drops of Kovacs. So we're going to do 4 to 5 drops. So make sure you're adding Kovacs reagent, and we're just going to add that to the surface. We're not going to shake it. Just add it to the surface, and you're going to look for a red band to appear.
Yes.
If my red band appears at the surface, that means my organism was able to break down tryptophan.
So that's equal.
Okay? If I don't see a red band, then my organism was not able to break down tryptophan. So 4 to 5 drops
Like a little bit red.
with Kovacs, and we're looking to see if we're able to break down tryptophan. So if I break down tryptophan, I'm able to create an indole reaction, which is going to again be an acidic shift. So, do we see some red developing?
Yeah.
Yeah, who's turning red?
E.
E. coli, good. So E. coli is positive for motility. E. coli is also positive for my indole, okay? So this is kind of nice. I'm able to key out my organisms here pretty quickly. Okay, the last one is the ornithine test. Now the ornithine test is really interesting. So we have our amino acid in here that's ornithine, and with our slant, or with our D, what color did they start?
Yellow.
Were they yellow when we started,
Like-
or were they purple?
Oh.
They were purple, yeah. So this is a weird one. So we start purple, and if we're positive, we end purple. That's really weird, but let me explain what's happening, because it's kind of cool. So, for my organism to create and activate decarboxylase enzymes, I have to have an acidic environment. That's weird, right? So remember how we talked about how some enzymes get turned on at specific environments? So the way this works is in my media, I have dextrose sugar. That's just like glucose, okay? My organisms start eating the sugar. If I eat sugar, what kind of pH do I create? Acidic. So now I create an acidic environment. Now, if I have decarboxylase enzymes, it's acidic around me, so they get turned on. And so then I start eating the proteins, and if I'm decarboxylating, what happens to my pH?
It increases.
It goes up. So I get a drop and then an increase. So this will throw a lot of first-year students, because if they read these tubes out at 24 hours, everybody's yellow. That means that I have pH dropping. But is that what I'm looking for when I'm looking for proteins? No. I have to let it sit for another 24 hours to see if my pH goes back up. all right, see if I recover. So it's a weird one because we start purple and we end purple if we're positive. Kind of an oddball. Who is showing a purple coloration in their tube still?
E. coli.
E. coli. Look how nice I am to you guys. With this test, E. coli was positive for everything. See, I try to be nice sometimes, right? All right, so again, everything was positive for E. coli on this test. Now, what that means then is if I'm purple, I'm taking ornithine and I'm breaking it apart. And the next thing I break it down into is putrescine. Ooh. Does that sound very nice? Yeah, you think of putrid, right? Sure enough, this putrescine will also break down eventually into cadaverine. Oh, it even gets worse, right? So, have you ever smelled something that's decaying or rotting? That's what that is. It's as when we die, we start to break down, sugars break down, bacteria break us down, we get a little bit acidic, and as soon as we get acidic, decarboxylase turns on, and we start having weird protein reactions. So some of the smells associated with decomposition come from this reaction right here. So, just a little interesting side note. So go ahead and record your results. Let's take a peek, because this is all of our main stuff, at our acetylene, our VP, and see if we have any color developing. How's it doing? Anything yet? Nope. Okay, so there's coli is out of class. Oh, so coli is on. Type should. That's the type right there. I'm not seeing too much yet. Anybody seeing club still? So we're just looking at club at this point. Right. Yes. That's your club. So we'll let them sit a little longer. See if you fix that. If not, I'll show you a picture.
Did you guys add your
See,
methylene already?
sometimes-
No, we didn't do it because we didn't really understand it, and we didn't think we did it right.
She said to do it, and I was like, wait.
We did the VP one, but we didn't know which one to add that to.
Yeah, that's why
So I didn't want to
I
ask.
feel like she's going around checking. And
If it sounded like just one, but we did it on both, didn't we?
Yeah. But how we're supposed to know if it's both because it's E. coli. Like they're two different bacteria.
But then the red, she's
But the thing is that we have enough to do
Do y'all
more.
see some color pretty? She's starting to get a little bit of a tannish pink line right here. It's very, very faint. I'm gonna have her go it sit a little bit longer. But we're starting to get a little peachy coloration. That's what we're looking for, that peachy pink color. So we're starting to get there. We'll let it sit. Y'all do not have to wait for this reaction. Like you can stay here, work on your notes, you can keep an eye on it. But if you are going to leave to go to a study with the water, it's okay. I'll show you a picture later and like what it looks like. All right? 'Cause it's very faint. It's kind of like you can squint at it.
We were supposed to add this. This is what I dropped into one of them. So we added.
I thought I knew, but I guess she explained these ones. I didn't understand that. I did my questions if you want to
We
repeat.
can just redo it. We can ask them and just redo it.
Yeah. But we're gonna have to wait longer for it. Like, if you want to do it, you can.
I should take a picture.
I'm gonna take a picture of it. Question there. I'm gonna ask her about this later because I didn't understand.
Okay, all right. So check it out. We got a strong reaction going on over here with our VP. So a lot of times this can have a difference with how much bacteria was in here to react with the VP reagents. This is looking great. This is probably one of the best reactions I've seen. So kudos. You did a good job. All right, all around. So we're seeing that it's definitely getting that rosy pink. Which one is that? The methyl red. So that was at the beginning with the VP drop. So you did your aliquots, right? So then with the tubes that have the most media, you're going to add 5 drops of methyl red to this guy and 5 drops to this guy.
and then these are just to change them.
Right, so these ones you added VP to. So we're supposed to see kind of a rosy color change in our VP.
It looks like rosy.
It's starting to get there, yeah. When you hold them up against the E. coli, it's starting to get a little pinker, right?
Nice glasses.
Yeah, so the club seal should get kind of a rosy color.
How's your BP looking? Is it looking more
We
rosy yet?
lost stuff on it.
Oh, okay, no worries, no worries. Just make sure you look at somebody else's results and then
Imagine you're the
No, probably.
Look at me, look at me. I literally guessed 2 on all. Not even joking, I guessed 2 on all of them.
I should do this all along, you know?
Every day.
I can see me for something now. Honestly. I don't like that one.
Do it. Honestly, can I check for you?
Yeah, you're fine. Change that.
So right now we're just waiting for the
I
MRI.
am the 15. So, that's okay.
Yeah.
It's fine.
Yeah. I go back tomorrow.
You did really good.
Wait, how did you do?
We got a really good score.
Let me see.
I got 28.
That's good. You got higher than me.
I don't know how I did it, honestly. I was so tired.
That's good though. Let me see.
Dude, I started panicking because I thought like everyone was done, and I looked up and I saw everyone was done, so I was like, oh no, I just hurried up.
That's good. How much time do you have to complete your quizzes?
Do you
It
have?
gave me 30 minutes.
We only get 15.
So yeah, I get like double. So just
That's good because I needed that extra time.
honestly, it's just like looking to LAP. I feel
But
like
what do I
anyone,
do?
you just like schedule an appointment to like get tested, and then depending on how well you do, you like.
Oh,
I feel like, cause like I had a classmate.
but you
who,
guys don't come back to class. Why?
Yesterday, cause I just did. I just wanted to go home.
Good job.
Do it.
Thank you. But
So once we're done reading everything
just
out, you can put your old tubes to the side. If you want to wait a while on the BP, you can. You don't have to. I will show you a picture of class later.
How about the oxidated?
Just knowing mostly that the MR and B
wait,
can
so
react to
all the data that we collected on Monday and Wednesday is in the group chat.
The labs?
Yeah, like up for this.
Well, I did all the labs and I put them on a slide for you. Not slide, on a Word doc. So like I wrote the results and everything.
And then if
Oh,
you're
for 12.
on anything, come see.
For 8
The one
to
that you
12.
just sent?
Yeah.
Oh,
So you should be good, and then I'll input the MRVP later.
More than I am.
Our lab exam is next Monday. This Monday coming up. And then it's our lecture exam.
Like a week from this.
yes. Which I'm gonna be in Mexico on Wednesday. Wednesday to Wednesday.
You're always doing something.
I know. It's like the worst
So it's
semester
our
to be
lab
doing
and
things.
then lecture.
Huh?
Where in Mexico are you going?
I'm going to Coahuila, Torreon, Coahuila.
Is that north or south?
It's more inland. It's like
But is it north or south?
nowhere. Nowhere. It's like closer to the United States.
So north.
So north, yeah. North and inland.
Because the south is looking rough.